All right, so this question kind of combines a little bit of what we've seen already. Here we've got a made-up example with geckos. Geckos with the dominant big S allele have stripes. Little s individuals are solid green. So let's start laying this out maybe. So big S something, stripes. And I'm going to do a funky little s so that I know it's lowercase, solid green. And here we've got a different locus affects stripe color. So true breeding red striped geckos and true breeding yellow striped geckos produce orange striped offspring. So again, not a terribly complex question to figure out, I guess, the inheritance here. If we have one gene called C here, and crossing red by yellow gives us orange, we've got a blended color there. That sounds like, think about it. Yes, I would say incomplete dominance. Good answer. And then we've got this other, so we've got stripes or no stripes, and then the color of stripes. So we've got a little bit of a situation where one gene is going to tell us whether we see the phenotype of the other gene. So here we had, possibility of red stripes, yellow stripes, and orange stripes. C-R-C-Y, orange. So maybe a little bit more complicated here. Two genes, only two alleles each, so maybe a little more manageable. So the relationship between the S gene and the C gene, we see another one of these kind of gatekeeper relationships. So the S gene has to be at least one dominant allele for S before we can see any of the stripe phenotype. I don't know if that was clear in the question, but that's what I intended here. So this is a case of epistasis. So working forward here, let's say we have two orange striped geckos that could have a solid green offspring. So is this possible? We got orange by orange, orange stripe, I should say. Produces a solid green offspring. So orange we determined was CRCY. And what do we know about the S gene if a gecko has orange stripes? We know that it has at least one big S. So I'm going to draw a big S blank there. Is it possible for us to have somebody in the offspring who is little s, and then it doesn't really matter about the C alleles? Why, sure, that's possible. So if this individual was heterozygous and this individual was heterozygous, then they could both give a little s to the offspring, and that would be possible. So CRCY for both parents and heterozygous for the S gene for both of those. All right, so let's say we had two of those. What are the chances that they produce orange striped offspring? Good question. All right, possible gamete genotypes. This one's kind of handy. I guess we can do a gamete square. And here we would have the same genotypes for both parents. So once we solve this for one, we'll have it for the other. So big S can be with CR, big S can be with CY. little s can be with CR, and little s can be with CY. So good example here of why I need to distinguish my capitals from lowercase. I'm not always writing those uppercase s's very clearly as bigger, but I have them written differently so I can tell they're bigger. All right, so 4 possible gametes for each. Oh my goodness. Big S, CR, big S, CY, little s, CR, little s, CY, across the top. Big S, CR, big S, CY, little s, CR, little s, CY. Maybe we could save some time here. So the question of how likely they produce orange striped offspring. So I can kind of eyeball this here and say we've got a few cases where CR and CY would mix in together. So CR, CY, CR, CY, CR, CY, CR, CY. Depending on how you write these out, sometimes you get a nice pattern of how they distribute on the grid. CR, CY, CR, CY. All right, so I just did places where the R from the top would mix from the Y from the side, or the Y from the top mixes with the R from the side. And that was about half of the places on this grid. But we need something else to happen. We need there not to be the little s combination. So the little s combination would happen down here in this box where we have little s's on the top parent and little s's on the side parent. So those, in spite of having CR, CY in their C genotype, would not show that because they have this epistasis, epistatic S genotype. So we can rule those out. And at this point, we would have the right answer. These are all the ones that I wrote in this part of the grid have at least one big S. So 1, 2, 3, 4, 5, 6. And our total grid size is 16. So that's 6 out of 16. You could reduce that down. Reduce it down to 3/8. Let's see if that's an answer. Hey, look, it's a possibility. So 3/8. You could also get at this question using a more probability approach, but I think I'll probably do that in the probability lecture for us. Okay, so kind of coming back around, we'll see some examples of human pigmentation. I think this is a topic of interest to people and also one that has a lot of interesting genetic tie-ins. This is from a study that shows the contribution, kind of the relative contribution of a variety of genotypes to pigmentation. So not just the overall appearance of a person, but getting down into their tissues and these melanosomes, the parts within a cell where these melanin pigment molecules are being joined together. So cells that have more melanosomes are darker. These are skin cells. Cells without the melanosomes are lighter. And of course, we know how people can have a wide range from light to dark skin. This is showing kind of a cumulative effect of having genetic production of pigment based on 3 separate locations. So different genes in different parts of the genome, the MATP, NCKX5, OCA2. Depending on your genotype, so this is a nucleotide genotype here, the TT genotype gives a higher production of melanin, the CC genotype, a lower production of melanin, and CT genotype, kind of intermediate. So altogether, this one gene has kind of an incomplete dominance inheritance type, phenotype. But it's not the only gene contributing to skin color. So if you wanted to find the genetic cause for a person's skin color, you'd have to look at this gene and this gene and this gene, and possibly others besides. And then these two show that there's variation in the amino acids. So it's not just, it would be nucleotide changes that cause a difference in the amino acid sequence. So these letter abbreviations are actually amino acid abbreviations, but basically it's 3 locations in the genome, all contributing kind of in the same way on the range from no pigmentation to lots of pigmentation. So a person with really dark skin would have pigmentation here, and pigmentation here. A person with really light skin would have no pigmentation here, no pigmentation here, and no pigmentation here. And then the range of genotypes in between, being homozygous, heterozygous for any of these, would kind of incrementally add on a little more pigmentation to the overall pigment profile of those individuals. So here's one way to think about it. I've just kind of simplified it down into 3 genes with alleles. Anybody who's got a capital letter is contributing pigmentation. So kind of on an incomplete dominance. The number of capital letters in somebody's genotype is more or less an indication of their pigmentation. So this person has 3 capital letters. They would be kind of a medium-skinned phenotype. This person has 3 capital letters. They'd be kind of a medium-skinned phenotype. And the question of whether these people could have a child with something that maybe is a little counterintuitive for some people, have a child that's more pigmented than either of them is. So someone with darker skin than their parents is totally possible. So let me see if I can write these out. I have to refer back to that a couple times. All right, so left parent was heterozygous. I'm just going to do simple single letters here. So both heterozygous for A. I can't help myself. And then homozygous recessive or homozygous dominant for T. All right, so what are the chances that they would have offspring with big L, 2 big L's, 2 big A's, and heterozygous for T? L, L, A, T, T. All right, so 3 genes. I guess we could probably do that. It's not too complicated. Number of unique alleles here. So this is homozygous, or unique gametes, I should say. So this individual could have big L, big A, little t, or big L, little a, little t. They're only possible gametes. There's only one gene that's heterozygous here. This individual has 2 heterozygous genes. Big L, big A, big T, big L, little a, big T, little L, big A, big T, little L, little a, big T. 4 possibles. Kind of skipping the gamete squares. You could do that with these though. So our Punnett square, let's do this person across the top. L, A, T, L, A, T, L, A, T, L, A, T, shoop, L, A, T, coming from over here, L, A, T. And I wonder if we could cut to the chase here. So we need, let's see, I already wrote that on there. This kind of offspring. So 2 big L's and 2 big A's. So anybody who's got a little a is going to be ruled out. Two big L's. This person, so we got a little L, that's not part of the genotype. Little a is not part of the genotype. Little a is not part of the genotype. Little a is not part of the genotype. So we got, looks like one possible chance out of these 8 total with the big L, big L, big A, big T, little t. I guess maybe if you're curious, we could look over here and find somebody who is maybe the least pigmented possibility. So 1, 2, 3, 4, 5 pigmentation alleles in this person and 2 pigmentation alleles in this person from parents who were each, we have three pigmentation alleles in each parent. So just a nice combination of phenotype to genotype ratios, and what might seem a little difficult to track on the surface, you could get in there and maybe find the genetic predictions for the transmission of this phenotype. Now, skin color and eye color are among the features that have lots of genes responsible. So there's variation in the kinds of alleles that are present at the genes responsible. There's variation in the genes specifically that are responsible. And it's an ongoing question. It's not so simple as just, I have a certain color of eye, and you can tell me exactly what my genotype is. So eye color, very interesting kind of spectrum of genetic causes. This is from a study where they were quantifying, they put a number on how brown versus how blue somebody's eyes were, sort of an aggregate investigation based on number of points, like number of pixels in these eye images that were scored as brown versus blue. So things on the positive end were more blue than brown, and on the negative end were more brown than blue. And they were matching that with genotypes, and they found a couple places. These are actually SNPs, single nucleotide polymorphisms, that show a pretty good split between the presence of an allele and the score on the eye color. So individuals with the G allele scored way more on the positive end of this range, looking at blue eye color. People with the A allele very strongly, homozygous for A were very strongly in the brown color. Same thing here, individuals homozygous for T at this gene, this location, very strongly brown eye color, and then a little bit more likely to be blue eye color with the C allele there. So even in this study, 2 independent genetic causes that could cause a person to have blue eyes or brown eyes. And I don't know what the total number is. It's 5 genes, I think, at least, or five separate regions that influence eye color. So lots more investigating to be done on this one.